Good afternoon, students.
Good afternoon, ma'am.
কি হচ্ছে অনেকদিন পরে ক্লাস শুরু হতে ভালো লাগছে না খারাপ লাগছে? মনে হচ্ছে ব্যস্ত ছিলাম, তাই তো?
হ্যাঁ ম্যাম।
হ্যাঁ ম্যাম। এটা কার গলা? দীপনের গলা, তাই তো?
দীপেন ম্যাম।
আচ্ছা দীপেন, সরি। কি পড়িয়েছিলাম লাস্টে? কি পড়িয়েছিলাম রে, মনে আছে? মিক্সিং
ম্যাম।
আমি শেয়ার করি স্ক্রিন, তারপরে বলছি। দাঁড়া, এটা আমরা পড়ি, এটা আমি ছবিটা দেখিয়েছিলাম। মিক্সিং কি বলেছিলাম আমি? যে you can see the particles are segregated. The particles are segregated here. Segregated, sorry, G-A-T-E-D. The particles are segregated here. We can see the round shape particles are there and triangle shape particles are there, and they are completely separated. They are segregated. Due to mixing, what will happen? Due to mixing, I have told you two terms. Due to mixing, there will happen randomization. There will happen randomization or there will happen randomization or combination. There will happen randomization or combination. Combination of dissimilar particles. দাঁড়া, অনেকদিন পড়ানোর অভ্যাস চলে গেছে তো আমার, তো থেকে তাই একটু সময় লাগছে। Okay, so the particles are segregated here, this one and this one. Due to mixing, the particles are now randomized. So mixing occurs, randomization or combination of dissimilar particles within a system. So if anyone asks you what is the definition of mixing, we can say like this way: mixing is a unit operation. Mixing is a unit operation by which segregated particles or separated particles can be randomized or combined into a unit mass or unit substance. So if I ask someone what is the definition of mixing, what you will say? Just now I have told you the definition of mixing. So what will be the definition? Anyone from the group?
Hello ma'am.
বল।
বলছি ম্যাম, হ্যাপেন র্যান্ডমাইজেশন অফ কম্বিনেশন অফ পার্টিকেলের পর থেকে আর শুনতে পাইনি।
আচ্ছা, বাকিরা যারা শুনতে পেয়েছে, এনিওয়ান ফ্রম দ্য গ্রুপ? যে কেউ? I have told you mixing is an unit operation process by which segregated or separated particles are randomized or combined into a unit mass or a unit substance. That is known as mixing. এবার বলে দিয়েছি আরেকবার মিক্সিং এর ডেফিনেশন কি বলেছি? Anyone from the
Ma'am, mixing is
অনিকেত বল
হ্যাঁ ম্যাম
বল
ম্যাম mixing is a unit operation process by which segregated and or separated particles are converted into randomization or combination by a unit
আচ্ছা আর কনভার্টেড ইনটু এ randomized
or combinant particles
ওকে
by a unit mass
টু গেট এ ইউনিট মাস তাই তো স্বরূপ স্বরূপ কি বলেছি স্বরূপ আরেকবার বল
Yes, ma'am. Mixing is a unit process when segregated or separated particles are randomized or combined in a unit mass or
Yes.
in a unit substance.
Yes, the segregated or separated particles are randomized
Randomized or combined.
into a single mass. তাই তো
Yes, ma'am.
এখানে দেখা যাচ্ছে এগুলো পুরো segregated ছিল এখন আমরা বলতে পারি এগুলো randomized আছে next one I will go to this one here the another definition is written mixing is a unit operation process in which two or more components are treated so that one kind of component will be lying next to the other kind of component. You can say this one as well as you can say the previous one. The segregated or separated particles are randomized or combined into a unit mass. I have told you also that mixing are of two types. One is ideal mixing and another one is acceptable mixing. Yes, ideal mixing means what? For that ideal mixing I have shown you one picture. See, this is the ideal mixing. This is the ideal mixing. What is the ideal mixing? Here one particle is lying next to the another type of particle. One type of particle, this is rectangle, this is sphere or oval shape. We can see one triangle shape is lying next to the oval shaped particles. That means ideal mixing, what will be the definition? The definition will be one type of particle will be lying, lying will be lying next to the another type of particle so that we will get a homogeneous mixture. I am showing you what I have written here. You can see random mixing. No, sorry, ideal mixing. When each particle of one component is adjacent to the particle of another component is known as ideal mixing. Ideal mixing এর definition কে বলবে?
ma'am বলছি
বল।
when the each particle of the one component is the lying on the another particle
is lying next to the
another particle.
আচ্ছা another type of particle. another particle তো নিজের পার্টিকেলের পাশেও তো হতে পারে একটা ট্রায়াঙ্গেল আর একটা ট্রায়াঙ্গেলের পাশেও তো থাকতে পারে তাহলে হবে না another type of particle then it will be ideal mixing. Then I have told you that acceptable mixing but never, as we have, you know very well that generally we cannot give the examples of ideal gases but we can give examples of real gases. Like that you can never say that there is ideal mixing but you can say there is some real mixing. Real mixing are of or acceptable mixing. Acceptable mixing are of two types. What are the types? One is random mixing and another one is ordered mixing. One is random mixing, another one is ordered mixing. Before going into details, I will give you one example. Suppose, let me see your name, then I'll give you the example. Who is present today? Okay, suppose Abhas is making tea, and Abhas knows that how to make tea. He knows the procedure. So what he has done, he has taken a kettle, then he has taken water, then he has given 2 spoons of tea, and he has started to boil the water. And then what procedure he has followed? He has to make 4 cups of tea. Then he has taken 4 separate cups. And in each cup, she has taken 1 spoon, 1 spoon, 1 spoon of sugar. So in every cup, he has given 1 spoon of sugar. Then when the boiling is over, then what he has done? He has strained the tea into each cup, and he has started to mix. He has started to mix with a spoon. But he is very lazy now. So what he has done, for first cup, he has started for one time. For second cup, he has started for single time. For third cup, he has started for single time. And for fourth cup, he has started for suppose 3 or 4 times. He has started very well. But for the first cup, second cup, and third cup, he has not started very well. খুব ভালো করে না নাড়িয়ে, চামচটা খুব ভালো করে না নাড়িয়ে চা দিয়ে দিয়েছে খেতে. So the person who is taking the first cup of tea, he may feel that okay, tea is good, but sugar is less. Second person may feel yes, the tea is good, but maybe sugar is little bit less. But the fourth person who is taking the tea, he may say that no, sugar, the tea is good and the sugar is also appropriate. Because for the fourth one, Abbas has given some more effort. He has studied for 2 times, 3 times he has studied. But for the first cup to third cup, he has started for single time. So actually what he has done the random mixing. Random mixing means some sort of mixing is there, but that is not uniform. Sugar is mixed in every cup, but for the first cup, second cup, and third cup, sugar has not been mixed very well. So random mixing, Abbas has mainly done the random mixing. Random mixing definition is some sorts of mixing is there, but homogeneous mixture you will not get. Now another one is ordered mixing. Ordered mixing এর এক্সাম্পল আমি বলি। অর্ডার্ড মিক্সিং কিভাবে হয়? অর্ডার্ড মিক্সিং এর জন্য আমি বলছি Abdul. Now second one, now Abdul is making tea, and he has followed the same procedure. He has followed the same procedure as Abbas has followed for making tea. But for the last one, that means when he has taken 4 cups and he has taken 1 spoon of sugar in each cup, he has started very well. For first cup, he has started for 3 times. He is giving some time. He is giving some pressure. He is giving some external force. So he has started for 3 times. For second cup, he has started for 3 times. For third cup, he has started for 3 times. And for fourth cup, he is starting it for 3 times. That means he has given some external force, he has given some extra force, and that one, when the person will take the tea, for the person who is taking the first cup of tea and for the person who is taking the fourth cup of tea, everybody will say no, that the tea which has been made by Abdul, it is good and it is having same sugar and it is feeling, that means the same amount of sugar is present throughout the tea. This one is the ordered mixing. So what will be the definition of ordered mixing? Ordered mixing means to do the ordered mixing, you have to apply external force, you have to give some extra efforts. For to get ordered mixing, you have to give some external force, you have to give some extra effort, so that the mixture will be homogeneous, and the homogeneity of the mixture will be very close to the ideal mixture. That means through ordered mixing, the mixture which you will get, that homogeneity will be very close to the ideal mixture. So who will give me the definition of random mixing and ordered mixing? At first tell me what will be the definition of random mixing, and then tell me what will be the definition of ordered mixing. Who will say the definition of random mixing? Who will say the definition of random mixing?
Sir, ma'am, bolbo.
Bol Shoro.
When each particle of component is not adjacent to the particle of another component, but some sort of mixing is there, is called random mixing.
Okay, that means some sort of mixing is there, but it is not homogeneous throughout the mixture. Right?
Yes, ma'am.
And what is the acceptable mixing? Ordered mixing?
Some external force is required so that mixture will be homogeneous.
Homogeneous, and it will be very close to the hero. It will be very close.
Ideal mixture.
It will be very close to the ideal mixture. তার মানে আমরা যদি কোনো এফোর্ট না দিয়ে ছেড়ে দিই, আমরা মোটামুটি র্যান্ডম মিক্সচার পাবো। কিন্তু যদি আমরা এফোর্ট দিই, অর্ডার মানে এক্সটার্নাল ফোর্স দিই, then the mixture which you will get, that will be very close to the ideal mixture. Next one, application of mixing. কি applications আছে? What are the applications of mixing? Now I will tell you. Suppose Abhijit is working in a company and he is preparing the tablet. But what he has done? He has done the random mixing rather than ordered mixing. He has done the random mixing and he has prepared the tablet. So what will happen? It may happen that if he has prepared, suppose he has prepared 100 tablets, and if he has prepared 100 tablets, then maybe 10 tablets will get the whole active ingredients, and remaining 90 tablets may not get the active ingredient. So the person who is taking that 10 tablets, they will get the overdose, and the remaining persons who is getting the 90 tablets without any active, they will be taking the tablet without any active excipient. That means they will use placebo. So to maintain content uniformity of tablet, capsule or any kind of pharmaceutical formulation, we need mixing. Usually mixing is proper na ho. If you are not doing the mixing in a proper way, if you are not doing the mixing in a proper way, then what will happen? Then some tablets may be prepared with higher amount of active, and some prepared may be devoid of any kind of active. To overcome this kind of problem, mixing is very much needed in pharmaceutical industry when you are preparing any kind of tablet, capsules, or any kind of pharmaceutical formulation. Now the second point is in case of potent drug. Potent drug means what? Potent drug means where low amount of the drug is sufficient to give any therapeutic benefits. Suppose you are preparing, suppose you are mixing 100 mg of tablets, 100 mg of active with 100 grams of excipients. So the amount of drug is how much? 100 mg. And amount of excipient is how much? 100 gram. See, this is mg. Drug is mg and excipients is gram. 100 mg is mixed with 100 gram of excipients. And why you are taking the 100 mg of drug? Because drug is very potent. Potent means low dose is sufficient to give therapeutic benefits. So when you are working with very potent drug, you have to be very cautious so that the mixing is happen so that you can do the mixing very well. Otherwise, some tablets will be prepared with drug and some tablets will be prepared without any drugs. And as the dose is potent, as the drug is very potent, so the patients who will be taking the tablets with drug, they may feel some serious side effects or serious toxic effects. And the patients who are taking the tablets without any kind of active, they may not get any therapeutic benefits. Actually, they will be taking placebo. They will take the tablet as a placebo. Now, number 3, to get true solution. When you are mixing two miscible solvents, they are generally mixed very easily. So it's not a problem for two miscible solvents. But number 4, to get emulsion. How the emulsion is prepared? Emulsion is generally prepared by forcefully mixing with water with oil. That means oil and water both are immiscible, and you have to mix oil with water or water with oil to get the emulsion. And this time in your lab, generally you prepare emulsion by taking sometimes by taking mortar spatula. And when you are using the spatula mortar, what you do, you give some external force, you stir it for several hours, several minutes, so that you get emulsion. So to mix two immiscible solvents, you have to give external force, you have to give extra efforts so that you can get homogeneous emulsion. Next one, to enhance disintegration and dissolution properties, we have to mix disintegrating agent properly. কেউ ট্যাবলেট বানাচ্ছে, ট্যাবলেট বানানোর জন্য স্টার্চ একটা ডিসইন্টিগ্রেটিং এজেন্ট, ট্যাবলেটের মধ্যে স্টার্চ দিয়েছে কিন্তু বাট দে হ্যাভ নট মিক্স দ্যাট স্টার্চ ভেরি ওয়েল। অনেকে কি করলো, যে যারা ট্যাবলেট বানাচ্ছে তারা স্টার্চ নেবে কিন্তু স্টার্চটা খুব ভালো করে মিক্স করলো না, তার ফলে কি হবে, সাম ট্যাবলেটস will be having more amount of starch and some tablets may not having any amount of starch. So the person who will take the tablet without any starch, that tablet will remain intact and it will come out without disintegration. And the person who will take the tablet with higher amount of starch as a disintegrating agent, that tablet will disintegrate very quickly and they will get the therapeutic benefits. So when you are mixing disintegrating agent in tablet, you have to be very careful, you have to mix it properly. Then last one, another one, to bring a physical change. Suppose you are mixing color, you want to make some red colored tablets, but you are not mixing the red colored or red dye properly. Then what will happen? Then it may happen that out of 100 tablets, 10 tablets will be very colorful and other 90 tablets will not having any color. Why? Because you have not mixed the coloring agent properly. That's why. And next one, to promote a chemical reaction, mixing is very important. So what are the applications of mixing? Anyone from the group? Anyone from the group
Yes,
can
ma'am.
describe?
Yes, ma'am.
Yes, ma'am.
Hare Krishna.
Yes, ma'am.
Hare Krishna. One by one. Hare Krishna. The first
Yes,
one.
ma'am. To make a tablet or capsule, to maintain content uniformity of tablet mixing is needed. তারপরে 2 নম্বর হচ্ছে যে in case of potent drug, if large amount of excipient is mixed with a low dose of drug, we need mixing. তারপরে to get true solution, mixing is important. To get homogeneous emulsion, we need mixing.
We need mixing. We need to mix two immiscible solvent. Okay. Then Hare Krishna, continue.
Yes, ma'am.
Then
To
other.
enhance
Okay.
to enhance disintegration and dissolution properties, we have to mix disintegration agent properly
You have to explain this point.
to promote a
You have to explain this one. Disintegrating and dissolution properties, you have to explain this one. কিভাবে এক্সপ্লেন করবে? এক্সপ্লেন না করলে তো হবে না। এক্সপ্লেন
কোনটা ম্যাম?
এই যে যেটা বলে নাম্বার 5 টু এনহ্যান্স ডিসইন্টিগ্রেশন। অন্য কেউ বাকি হরকৃষ্ণ ছাড়া, ইফ এনিওয়ান ফ্রম দ্য গ্রুপ?
স্টার্চ নিতে হবে ট্যাবলেট বানানোর সাথে।
দিনেশ কন্টিনিউ। নাম্বার 5
ম্যাম স্টার্চ নিতে হবে ট্যাবলেট বানানোর সাথে, ওটাই
না। দেন এটা ব্যাস, তারপরে কি হবে? স্টার্চ নিয়ে, স্টার্চ কেন দরকার?
ডিসইন্টিগ্রেটিং এজেন্ট।
অভিজিৎ বল।
স্টার্চটাকে প্রপারলি মিক্সিং করতে হবে, ওটা ডিসইন্টিগ্রেটিং এজেন্ট, তো ওটা মিক্সিং করলে যে রেশিওটা যেটা সেটা যেন ঠিক থাকে, মানে কোনোটাই 10% কোনোটাই 90% না, প্রপারলি সব কিছুতেই যেন প্রপারলি থাকে, সেই কারণে।
আচ্ছা, যারা স্টার্চ পাবে না, what will be the problem? অভিজিৎ, যদি কোনো ট্যাবলেট স্টার্চ না পায় কি প্রবলেম হতে পারে? Some tablet is not having any amount of starch, then what will happen? বল, যদি স্টার্চ না পায় তাহলে কি প্রবলেম হবে অভিজিৎ?
ওই ডিসইন্টিগ্রেশন যেটা, ওটাতে টাইম লাগবে অনেকটা।
হ্যাঁ, ওটা অনেকটা টাইম লাগবে, ডিসইন্টিগ্রেশন হবেই না, ইনট্যাক্ট হয়ে ট্যাবলেট বেরিয়ে যাবে। আর যেটাতে স্টার্চ বেশি থাকবে সেটার কি প্রবলেম হতে পারে?
মানে বেশি হবে
ওটা বেশি হবে। ওটা কি হতে পারে? অনেক সময় হয় কি, কোনো ট্যাবলেটস আমরা বলেছি যে সেটা ইন্টেস্টাইনে গিয়ে ডিসইন্টিগ্রেট হোক, এরকম আমরা ভেবেছি, কিন্তু স্টার্চ আমরা খুব বেশি দিয়ে ফেলেছি। বেশি দিলে কি হয়, ওটা তো খুব তাড়াতাড়ি ডিসইন্টিগ্রেট হয়, তার মানে স্টমাকে এসেই হয়তো তাড়াতাড়ি ডিসইন্টিগ্রেট হয়ে গেল। সাপোজ উই ওয়ান্ট টু dissolve or disintegrate the tablet in intestine only, but as the tablet is having more amount of starch, it may happen, starch at a disintegrating agent, it may happen that when the tablet will reach to stomach, it will disintegrate quickly. So instead of reaching to intestine, it will disintegrate in stomach itself, which is not desirable. That's why you have to mix the disintegrating agent within the tablet, within the powders or granules very well. Otherwise, what will happen? Some tablets may get more amount of disintegrating agent and it will disintegrate very fast, and some tablets may not have any disintegrating agent, and as a result, what will happen? As a result, it will remain as a intact mass. It will remain as an intact mass and it will not disintegrate. আর যদি ট্যাবলেট ডিসইন্টিগ্রেট না হয় তাহলে যে অ্যাক্টিভ ইনগ্রেডিয়েন্ট, দ্যাট উইল নট, ওকে। অ্যান্ড আরেকটা কি হচ্ছে, যদি ট্যাবলেটস না স্টার্চ পেয়ে থাকে তাহলে ওটা ডিসইন্টিগ্রেটই হবে না। নেক্সট ওয়ান বলেছি ফিজিক্যাল চেঞ্জ। ফিজিক্যাল চেঞ্জের জন্য কি বলেছি? কে বলবে এটা? কালারের কথা কি বলেছি? এনিওয়ান ফ্রম দ্য গ্রুপ? আর কেউ গ্রুপ থেকে? কালারিং এজেন্ট যদি প্রপার না হয় তাহলে কি হবে? some tablets may be very colorful and স্নেহাশিস।
yes ma'am, যদি ম্যাম প্রপারলি মিক্সিং না করা হয় তাহলে কালারিং এজেন্টটা কিছু ট্যাবলেটে 10% or 20% অফ ট্যাবলেটে বেশি কালারফুল হয়ে যাবে, and some less amount of coloring agent are containing the,
and some amount of the tablet, some tablet may be of
colorless.
Yes, some tablets may be very colorful and some tablets may be colorless. Right? And to promote a chemical reaction, সেজন্য আমাদের কি করতে হয়? মিক্সিং ইজ ভেরি ইম্পর্টেন্ট। তোরা যখন অ্যানালিসিস ল্যাব করিস, অ্যানালিসিস ল্যাবে মিক্সিং করার জন্য কি ইউজ করিস? অ্যানালিসিস ল্যাবে যখন তোরা কোনো ল্যাব করিস, that time you have to use glass rod. Why do you use glass rod? মিক্সিং করার জন্য। and analysis lab এ তুই গ্লাস রড ইউজ করছিস, আর কোনো ল্যাবে তো গ্লাস রড এইভাবে ইউজ করতে হয় না, বাট ইন অ্যানালিসিস ল্যাব ইউ আর ইউজিং গ্লাস রড। why you are using glass rod? Because glass rod is needed for mixing, and that mixing helps to promote chemical reaction. So these are the applications of mixing. Then types of mixing, আমি বলেছিলাম কি কি types of mixing আছে, solid solid mixing, solid liquid mixing, আর বলেছিলাম liquid liquid mixing। তিন ধরনের মিক্সিং এর কথা বলেছিলাম। লিকুইড মিক্সিং, সলিড মিক্সিং এর ডিফারেন্স বলেছিলাম। কি বলেছিলাম মনে আছে কারো? সলিড মিক্সিং,
Yes, ma'am.
Yes, ma'am.
এই তিনটে তো পরিচিত নাম। সৌরভ, এটা বলেছে অনিকেত, একটা ঐশিকা বোধহয়, বাকিরা ঈশিকা, বাকিরা
ম্যাম বলবো?
সৌরভ পাত্র বল।
সে সলিড মিক্সিং কাজের আগে ওই ড্রাই পাউডার
ডিফারেন্স বলেছি আমি বলেছি সলিড সলিড মিক্সিং আর লিকুইড লিকুইড মিক্সিং, যেটা তার ডিফারেন্স বলতে বলেছি। হোয়াট ইজ দ্য ডিফারেন্স বিটুইন সলিড সলিড মিক্সিং এন্ড লিকুইড লিকুইড মিক্সিং? সৌরভ পাত্র, সৌরভ পারবে, সৌরভ বল।
সলিডটা মনে আমি সলিড
আচ্ছা আমাকে বল কোথায় এনার্জি বেশি লাগবে? সলিড সলিড মিক্সিং করতে বেশি এনার্জি লাগবে না লিকুইড লিকুইড মিক্সিং এ বেশি এনার্জি লাগবে? Where do you need more energy? Solid solid mixing need more energy and liquid liquid mixing needs lesser amount of energy. Now tell me, which one is quick? Which one is the quickest process?
liquid
liquid is the quicker one than the solid solid. কোথায় ফ্লো কারেন্ট দেখা যেতে পারে?
লিকুইড।
আচ্ছা, আর আরেকটা বলেছিলাম যে আরেকটা বলিনি আমি, আরেকটা হচ্ছে যে সলিড সলিড মিক্সিং এর জন্য ডিফারেন্ট কাইন্ডস অফ ইকুইপমেন্টস আর अवेलेबल, বাট ইন কেস অফ লিকুইড লিকুইড মিক্সিং জেনারেলি মিক্সিং ট্যাঙ্ক ইজ ইউজড and different kind of impellers are used. Impellers মানে স্টারার আর কি যেগুলো। তাহলে কি কি বললাম টাইপ অফ সলিড সলিড মিক্সিং এন্ড লিকুইড লিকুইড মিক্সিং এর ডিফারেন্স? সৌরভ পাত্র ছাড়া বাকিরা কি কি বলেছি?
সলিড সলিডটা দেরি লাগবে,
সলিড সলিডে মানে
রাহুল বল। হরকৃষ্ণ বলেছে, রাহুল বল।
সলিড সলিড মিক্সিং এর ক্ষেত্রে মোর এনার্জি আর ইউজড, আর লিকুইডে কম, আর
ইউজড ইয়েস,
আর সলিড সলিডটা এটা মানে বেশি টাইম লাগবে, আর লিকুইড লিকুইডটা এটা কুইক মানে তাড়াতাড়ি হয়ে যাবে।
ওকে, এন্ড অ্যানাদার ওয়ান ফ্লো কারেন্ট কোথায় দেখা যায় রাহুল?
ফ্লো কারেন্ট মানে লিকুইডে
একটা ফ্লো আছে, লিকুইড মানে ফ্লো থাকবে, ফ্লো কারেন্ট ইজ সেম, আর এখানে ফ্লো কারেন্ট দেখা যায় না। আরেকটা কি বলেছি? ডিফারেন্ট কি বলেছি?
ডিফারেন্ট ইকুইপমেন্ট
করা হয় সলিড মিক্সিং এ।
Different equipments are available for solid solid mixing, but for liquid liquid mixing generally mixing tank is used with different kinds of impeller, with different types of impeller আমি পরে আরো ডিটেইলস এ বলে দেব।
ওকে ম্যাম।
This one আচ্ছা, ordered mixing কার আন্ডারে আসছে?
ম্যাম
acceptable mixing এর আন্ডারে একটা হচ্ছে random mixing, একটা হচ্ছে ordered mixing. Ordered mixing can be done by three ways. One is mechanical means, one is adhesion means, and another one is coating means. Mechanical means, adhesion means, coating means. So by these three ways you can do the ordered mixing. First I will show you adhesion means of order mixing. At first see the picture very carefully, then try to understand what they have done or what they want to say. Then clarify this to me. At first see very carefully, you can see there are some fine powders of brown colors, there are some coarse powders, light yellow colors. Then they have done some ordered mixing by adhesion means. Is there anyone who can say me the steps how they have done the ordered mixing? You have to draw the picture in your copy, in your notebook. At first you have to draw the picture in your notebook, then This is the second means of ordered mixing. As I have told you earlier that there are three means for ordered mixing. First one is mechanical means, second one is addition means, third one is coating means. Now I am telling you the addition means. Mechanical means I will come later. At first I will tell you the addition means. See the picture. And is there anyone who can tell me the steps one by one? What actually they are doing? Anyone from the group, anyone? What they are doing? Just one by one step. Others will write.
Yes, ma'am.
স্বরূপ না, স্বরূপ বলেছে, বাকিরা? Others. স্বরূপ ছাড়া বাকিরা কে? এই স্টেপস গুলো কে বলবে? দিনেশ না, দিনেশ বলেছে। আর কে? স্বরূপ, দিনেশ ছাড়া? Who will tell me the steps? Okay, there is no one. Dinesh, continue. Okay, write down. At first, fine powders have to be mixed with coarse powders. Fine powders have to be mixed with coarse powders. You should write down. I won't give you note for this one. You just write it down. At first, fine powders have to be mixed with coarse powders. Fine powders are present. Fine powders are present in. Fine powders are present in. কি ফর্মে আছে? Fine powders are present in. Fine powders are present in. কি ফর্মে আছে ফাইন পাউডার গুলো? Fine powders are present in. কিভাবে আছে ফাইন পাউডারস গুলো? Agglomerated or deagglomerated? কি ফর্মে আছে?
agglomerated
Fine powders are present. Fine powders are present. Yes, fine powders are present in agglomerated form. So deagglomerations of fines. So deagglomeration of fines is to be done. Deagglomeration of fines is to be done. Deagglomeration of fine is to be done. Deagglomeration of fines is to be done. Next point, in next step, fine particles will be bonded to the coarse powder. Fine particles will be bonded to the coarse powder. And we will get ordered mixing of adhesion types. We will get ordered mixing of adhesion types. So now we will explain what is the adhesion means of ordered mixing. কি বলেছে আরেকবার বল আমাকে, যেগুলো বলেছি পয়েন্ট গুলো। Anyone from the group? যেগুলো লিখিয়েছি সেগুলো বল one by one.
Ma'am,
At first, fine powder have to be mixed with the coarse powder.
At first, fine powders has to be mixed with coarse powder. Okay.
Fine powder at present in the agglomerated form, so deagglomeration of fine is to be done.
Okay, তার মানে ফাইন পাউডার গুলো আমরা আগে দেখছি কি agglomerated ফর্মে আছে, so deagglomeration fines is to be done. Then
fine particles will be bounded to the coarse powder and
will be bounded বলছে আমি will be bounded will be bonded like will be bonded or attached will be bonded or attached
to the coarse powder
finally
and we will get ordered mixing of addition
we will get ordered mixing of addition means তাই তো we will get ordered mixing এখানে মিক্সিং হয়েছে কিনা এখানে মিক্সিং হয়েছে তো এগুলো আগে আলাদা আলাদা ছিল in the here we can see these are in separated form or segregated form when you are giving some external force or when you are applying force then what will happen these fine particles are attaching or binding with the coarse particles so we can see the randomization or combination of dissimilar particles brown particles with yellow particles randomization or combination of dissimilar particles and you will get ordered mixing now they are not present in a segregated form rather than they are combined now last one I want to ask you here do you think this kind of mixing will be very stable this kind of mixing will be very stable or they can anytime that bonding may be loose এটা কি খুব স্টেবল হবে বা যে কোনো সময় এটা খুলে যেতেও পারে এগুলো সরে যেতেও পারে do you think this one is very stable this bonding is very stable
no ma'am
this bondings may not be very stable তাহলে আমরা স্টেবল কিভাবে করতে পারি কিভাবে স্টেবল করতে পারি কোটিং করে coating means of ordered mixing you can see here we have taken ordered mixture you can write it down like this way we have to take ordered mixture this is the ordered mixture by addition means we have to take ordered mixture then we have to take ordered mixture by addition means we have to take ordered mixture by addition means then some coating solution is prepared then some coating solution is prepared you can see some coating solution is prepared then some coating solution is prepared then some coating solution is prepared and that coating solution is used to coat and that coating solution is used to coat ordered mixture and this coating solution is used to coat the ordered mixture number 3 ordered mixing by coating means are more stable ordered mixing by coating means is more stable ordered mixing by coating means is more stable than the adhesion means তাহলে এটা কি বললাম অর্ডার মিক্সিং এর কোটিং মিন্সটা কি বলেছি কি লিখিয়েছি just now I have told you how to do the ordered mixing by coating means anyone from the group
we have to take order mixture by addition means then some coating is prepared that coating solution is used to coat the ordered mixture
okay and this one তারপরে
ordered mixing by coating means are more stable than the addition means
okay আচ্ছা এরপরে কার ক্লাস আছে আমার ক্লাস কতক্ষণ রে 3:50 থেকে 4:40
yes ma'am
এরপরে কার ক্লাস আছে বুধা সাগরিকা ম্যাডাম নাকি
yes ma'am সাগরিকা ম্যাম
ম্যাম এখনো টাইম আছে 3 মিনিট আপনি নিন সাগরিকা ম্যাডাম 4:40 থেকে
আচ্ছা সাগরিকা ঢুকলো না এখনই মনে হলো সঞ্চারী
ম্যাম এখনো ঢোকেননি
আচ্ছা তাহলে আমি এন্টার করলাম যে ম্যাডামকে, বুঝলাম না তাহলে বেরিয়ে গেছেন ম্যাডাম বোধহয়।
ম্যাম আছেন ম্যাম আছেন,
আচ্ছা
আপনি নিন, আমি ওনাকে পরে হোস্ট বানাচ্ছি আপনারটা হয়ে গেলে।
আচ্ছা
ওকে ম্যাম।
দেন নেক্সট ওয়ান ইজ মিক্সিং ইন্ডেক্স। মিক্সিং ইন্ডেক্স কি? মিক্সিং ইন্ডেক্স এভাবে আমরা লিখতে পারি, মিক্সিং ইন্ডেক্স mixing index you can write this way. Mixing index is equal to standard deviation of, write in your copy, I may not save, standard deviation of random sample divided by standard deviation of samples, standard deviation of samples to be mixed, standard deviation of sample to be standard deviation of samples to be mixed. স্ট্যান্ডার্ড ডেভিয়েশনের কথা বলি আগে। স্ট্যান্ডার্ড ডেভিয়েশন যদি আমি জিজ্ঞেস করি যে কেউ ফার্স্ট সেমিস্টারে 100 পেয়েছে, সেকেন্ড সেমিস্টারেও 100 পেয়েছে, আর থার্ড সেমিস্টারেও 100 পেয়েছে, তার স্ট্যান্ডার্ড ডেভিয়েশন কত?
100।
কেউ ফার্স্ট সেমিস্টারেও 100 পেয়েছে, সেকেন্ড সেমিস্টারেও 100 পেয়েছে, থার্ড সেমিস্টারেও 100 পেয়েছে। এবার আমি বলতে চাইছি যে তার এভারেজ কত? এভারেজ 100
ম্যাম।
আর স্ট্যান্ডার্ড ডেভিয়েশন কত?
0
স্ট্যান্ডার্ড ডেভিয়েশন 0। আর কেউ যদি প্রথম সেমিস্টারে 100, পরের সেমিস্টারে 100 আর তারপরের সেমিস্টারে 90 পায়, তার স্ট্যান্ডার্ড ডেভিয়েশন কত হবে? 0 না একটু বেশি হবে?
10
নো, 10 হবে না,
0 থেকে বেশি।
0 থেকে বেশি হবে। ঠিক আছে, পরের দিন করব, 4:40 বাজে, পরের দিন বলবো আমি। থ্যাংক ইউ স্টুডেন্টস।
থ্যাংক ইউ ম্যাম।
ম্যাম।
ওকে।
Sagarika ma'am, you are co-host now, ma'am.
Hello.
Yes, ma'am.
Okay, thank you.
Okay.
Hello. Good evening, students. Can you hear me? Can you see the screen?
Yes, ma'am.
Yes, ma'am.
Okay. How are you all? How are you all?
Fine,
Not
ma'am.
bad.
Fine, ma'am.
You gave your exam, C1 exam. How did
Ma'am, your voice is breaking, ma'am. Ma'am, your voice is breaking.
Ma'am, I can't hear properly. Hello.
Everybody, please mute yourself. Sagarika ma'am, your voice is breaking.
Okay.
Yeah.
Now?
Ma'am, it's breaking sometimes.
Okay. Maybe due to the network.
Yeah, ma'am. Yeah. It's okay, ma'am. Now it's audible. Yeah.
Okay. So, in the previous classes, we studied animal cell culture. What is animal cell culture, right? and what are different types of cells we can grow. And we also study how to prepare media, right? So what are different types of medium? So how many different types of media we can use? What are different types of cells? Okay. And we also studied about contamination, right? What are different sources of contamination, how to identify the contaminations, all this out the growth kinetics of animal cells in culture. Okay, so what we are doing is first we will isolate the cells, right? So we have techniques for their growth.
Ma'am, can't hear you.
Ma'am, voice.
Ma'am, I think you are having some network issues, ma'am. You are not audible.
So media
Ma'am, yeah, the voice is breaking. Ma'am, you can rejoin if you want.
And they can you suggest? Okay.
Ma'am, yeah, if you want, you can rejoin, ma'am, because there is some problem. Ma'am, your sound is not coming. Hello, ma'am.
Hello.
Yes, ma'am. Ma'am, can you please move from the place you are right now?
Hello.
Yes, ma'am.
Is it audible now?
Yeah, it is very much audible right now. ma'am.
Okay. Okay. Yeah, let me start again. So in the previous classes, we studied about animal cell culture, right? So apart from it, we also studied about what are different types of cells we can grow in the laboratory and what are different media, right? Or how to prepare those media and how to isolate the cells, disaggregation of cells from the tissues by using physical, chemical, and enzymatic methods, right? So after that, we also studied about the contamination. What are different sources of contamination? How to identify the contaminations in the laboratory? Okay, all these things we discussed previously. So once after isolating the cells, we are keeping the cells in the medium, right? And we want them to grow in the media by using the nutrients which are present in the media. Okay, so once they start growing, what are different phases in their growth? We have to understand how the cells are growing properly or not. For that, we have to know the growth kinetics. Okay, so for this, we are discussing today to understand the growth of the cells. We are studying this growth kinetics. Okay, so now after isolation, we are keeping them in the media. So the environment is completely changed. Previously, they are in a different environment, and suddenly we are shifting them from the in vivo conditions to in vitro conditions, and we want the cells to grow by using the media which we give them, right? So there is a sudden change in the environment. So first what we have, the cells have to adjust with that environment, then only their growth starts. Okay, so let us see what are the different phases during the growth of the cells, okay, in the media, in vitro, in the laboratory conditions. So let us see the graph. So here we plotted a graph by taking time on x-axis, it is in days, okay. On y-axis, we took the cell density, that is number of cells which are present in 1 ml. Okay, so by taking this on y-axis, we are plotting the graph. When we plot the graph, you can see the graph which I plotted here. So you have different phases of growth. Initially, you can see the line is parallel to the x-axis, right? So that phase is called as lag phase. Okay, so the line which is parallel to x-axis, that is known as lag phase. The next phase which is present, which is moving up, right? So this one is known as log phase. Okay, so next is a stationary phase. This is a stationary phase, and then this is a decline phase. So we can study the growth of the cells in these different phases. One is lag phase, log phase, stationary phase, last one is decline phase. Okay, so first we will see what is lag phase. So the environment is new for the cells, right? So they are present in the in vitro environment and they are present in the media, and the environment is completely new for the cells. So the first, what it happens, the cells have to adapt to the new environment. Okay. So the first thing that happens is cell adaptation. Means the cells which are present in the media, they have to adopt. Okay, they have to adapt to different medium conditions, temperature, different enzymes, growth factors, different nutrients present in the media. So that phase is cell adaptation. So here what happens, cells are adjusting to the new environment, but there is no increase in the cell growth. Cell proliferation, cell growth is nothing but cells are dividing, cell proliferation. So that is not happening here in the lag phase. So what cells are doing? Cells are adapting themselves to the new environment and they increase in their size. There is no increase in the growth of the cells, means cell proliferation doesn't take place. At the same time, you can see there is increase in the cellular activities. So what are these cellular activities? Can anyone tell me? What are cellular activities that takes place inside the cell? Yes, anyone? I know it is a last class, but still I need some responses from you. So different cellular activities like respiration, right? So respiration happens inside the cell. At the same time, transportation, passive diffusion, active diffusion, several signals, all these are happening inside the cell. So the cells which are present in the lag phase, they have increased cellular activities. Not only that, they synthesize growth factors. Okay, so these growth factors will help in the next phase. Okay, so for growth of the cells, exponential growth of the cells, you require the growth factors, and these growth factors are synthesized in the lag phase. Okay, there is only expansion of the size. There is no increase in the cell growth, enhanced cellular activity, synthesis of growth factors, The cells will expand in their size and they adapt to the new environment. Okay, so these things will happen in the lag phase. Now, the duration of this phase, how long this phase continues, it is different. It depends on different factors. Okay, so what are different factors which decide the duration of this lag phase? First one is viability of the cells, means how many live cells are present. So when I add inoculum, is it audible?
Yes, ma'am.
Okay. So when we are adding cells to the fresh medium, you can't expect all the 100% cells will be surviving. No, all 100% cells won't survive. So there are some cells which die when you shift them from one environment to another environment, right? So we have to know what is the viability of the cells after seeding them in the new environment. Okay, so how do you know after seeding the cells? Seeding means you are adding cells to the media, fresh media after isolation, right? So after adding cells to the fresh media, you have to know how many cells survive this new environment and how many cells are dead, right? You have to know how many cells survived in this environment. For that, you can use trypan blue test. Okay, trypan blue is an dye. So in this test, what we will use, we will we will use generally 0.4% of trypan blue. Okay, so you will prepare this. First, you have to separate the cells by centrifugation. After that, you have to treat them with the dye. When you treat the cells with the dye, trypan blue dye, what happens? The time of contact should be only for 5 minutes. Okay, so the cells which cannot survive in the new environment, okay, so they will take the dye. The trypan blue dye, it permeates inside the dead cells. But this trypan blue dye cannot enter into the living cells. So the surviving cells or the living cells, they don't take the color. Okay, so using hemocytometer, you have to count the number of cells which are not stained. Okay, after counting the number of cells, you can calculate the percentage viability. That is, what is the percentage of the cells which you added into the media are survive, are in live condition. Okay, so that is known as percentage viability. How to calculate this percentage viability? That is equal to number of cells unstained. Means the cells which are not taking the dye, they are living cells or the live cells. Okay, you have to count that cells. So number of cells which do not take the stain, count them, divided by total number of cells present in that volume which you took for the counting. Okay, so after when you divide, you will get the percentage viability, multiply by 100. So the formula for percentage viability is number of cells unstained divided by total number of cells into 100 will give you the percentage viability or the live cells present in the flask which you are seeding. Okay, so what is the other factor that decides the duration of the lag phase? So the next factor is seeding density. Seeding density means the number of cells you are adding to the fresh media. How many cells? What is your seeding density? So generally, normally 10 power 4 to 10 power 5 cells per ml are added as a inoculum to the flask. Okay, so if you take 1 ml, it should contain nearly 10 power 4 to 10 power 5 cells होना चाहिए. Okay, so that is the normal inoculum which you take during the seeding. Seeding is adding cells to the fresh media. Okay, so when you inoculate, high density means large number of cells. When you inoculate, what happens? The cells, you will have large percentage of the cells which are living, right? At the same time, the capability of the cells increases, and they synthesize the growth factors as well as the enzymes which are required for the next phase for their growth. Okay, so what happens when you seed high density? The cells, the ability of the cells increases. As a result, we have more number of surviving or live cells. More number of cells are available. Now what happens? All will start working, and they synthesize the growth factors and enzymes which are required for the next phase. Okay, so next factor is energy charge. What is this energy charge? Can anyone tell me what is the energy charge? What is the main source of energy? Which molecule is responsible for the energy? Main source of energy? ATP. Yes. ATP is known as adenosine triphosphate. Okay. So this is the source of energy in the cell. For all activities of the cell, it requires energy, right? So by calculating the energy charge that is present in the living cells, that also decides the lag phase duration. Okay, so energy charge, it is a term that is related not only ATP but also adenosine diphosphate, adenosine monophosphate. All the three concentrations should be considered while you are calculating this energy charge. Okay, so the formula for calculating the energy charge is equal to ATP plus half ADP divided by total concentration of ATP, ADP and AMP. Okay, so when you do this fraction, you will get energy charge. What is the formula for energy charge? That is equal to ATP plus half ADP divided by ATP, ADP and AMP, the total. Okay, so when you solve this, you will get the energy charge. So energy charge, when you are calculating energy charge, you have to consider not only ATP, you have to consider even adenosine diphosphate and adenosine monophosphate should be considered. Okay, so why we are calculating this energy charge? What is the use of it? When you calculate the energy charge, the value helps to understand the viability of the cell population. How many cells are in living condition? You can know. Okay, so for healthy cells, generally the energy charge value is in between 0.8 to nearly 1. Okay, so 0.8 to 1. So healthy cells have this energy charge. The next factor is components of the media. The nutrients which are present in the media also affect the duration of the lag phase. Okay, so these are different factors which affect the duration of the lag phase. Understood? So next, coming to the log phase. So this is also known as growth phase or exponential growth phase. So here the cells will multiply, maximum they multiply. They become double. The number of cells in this phase, they become double the number, double their number. Okay. Not only that, during this phase, you can see the high metabolic activity inside the cells. Okay. So not only that, the metabolic activity of the cells is very high during this log phase. It is also known as logarithmic phase. Generally in this log phase, what happens? We can classify, interphase, mitosis, meiosis, interphase. Do you know?
Yes,
So
ma'am.
interphase is further classified into G1, S, G2 phase, right? So G1 is the first growth phase. S phase is the synthesis phase where DNA synthesis takes place, whereas G2 is the second growth phase after DNA synthesis. So the interphase is again subdivided into different phases. M phase is nothing but mitosis phase where the cell division takes place. If you take a normal human cell, okay, so generally the cell cycle is for 24 hours. The G1 stage will last for 11 hours. Okay. S phase for 8 hours, G2 phase for 4 hours. Okay. And M phase for only 1 hour. So total cycle is 24 hours. Okay. So what happens in this log phase? The cells have a very high metabolic rate. They double the number. To calculate after log phase, what is the concentration of the cells after multiplication? To understand, to calculate it, you can use this formula. N is equal to N naught into 2 raised to the power X. Okay, where N is the final concentration of the cells, N naught is the initial concentration of the cells, whereas 2 raised to the power X. X is nothing but the number of generations during the cell growth. Okay, so by using this, you can calculate what is the final number of cells after log phase. As I told you, log phase may cells multiply, they divide. Maximum multiplication of cells takes place. At the same time, the metabolic activities of the cells are very, very high. Okay, so after this log phase, another formula is to understand what is the growth rate. Rate kab use karte hai? With respect to which parameter? Rate. Rate means time. Yes. So we want to know what is the specific growth rate. Means in this time. Suppose the time is 15 minutes. In this time, 15 minutes of time, what is the growth of the cells? So with respect to time, that is specific growth rate. It measures the rate of increase of cell number. Okay. So with respect to time, that is represented by mu. By using this formula, you can calculate the specific growth rate of the cells. Okay, so mu is equal to dn divided by dt into 1 by n. N is the final concentration of the cells. Okay, so this is per hour. That's why it is given unit H inverse. Okay, so we are calculating the specific growth rate per hour. Okay, so for that, the formula is mu is equal to dn by dt into 1 by n. Got it? So specific growth rate helps you to measure the rate of increase of the cell number. How many cells are increasing in 1 hour? You can calculate by using the formula. Okay. So next important phase is lag phase. What is this? Sorry, stationary phase. So we finished the lag phase. Next is log phase. The third one is a stationary phase. So what is this stationary phase? In this phase, there is no further increase in the cell concentration. So cell division doesn't take place. Cell proliferation doesn't take place. So number of cells that are dying is equal to the growth rate of the cell. Okay. So this phase is called stationary phase. So contact inhibition. Can anyone tell me what is this contact inhibition?
Ma'am, contact inhibition is when one cell is growing and it had reached its limit, then same goes, same cells are there. If some cell is going, it's like it's inhibiting the other cell to grow beyond its actual shape. There is a limit after which the growth is stopped with by contact inhibition.
Why the growth stops?
Um,
Let me explain you. Okay, wait. Can you see the board?
Yes, ma'am.
Okay. So contact inhibition, it is a regulatory mechanism. Okay. It regulates the growth of the cell. Suppose this is your plate. Okay. In this you have different cells. Suppose you inoculated little concentration of the cells initially. So the cells are growing in the media. So this is filled with the media and they are taking the media and they are growing. Okay. So few cells are present in this plate. With the time, what happens? Cell divides. Okay. At one point, cell divides by using, they get nutrients from the media and they divide and they proliferate. Okay. So this contact inhibition is a regulatory mechanism. It controls the growth of the cell in a layer to form a monolayer. Okay. So the cells are growing, they form a monolayer. In one single layer, they grow. At some point of time, what happens? The space which is present in the plate is completely used up. So it is completely filled. There is no space at all, completely filled because of the proliferation of the cells. Okay, I'm not filling it completely, but to make you understand, I'm drawing to some extent. So like this, the space of the flask is completely filled with the cells. Okay, so now the cells are not having enough space to multiply, right? So as a result, what happens? One cell is coming in contact with the other cell, and there is no space for the proliferation. When they contact one signal with the other one cell with the other cell, what happens? That gives an inhibition signal for the growth, and the growth stops because of the contact. Okay, so contact between the cells will give a inhibitory signal for the growth, and as a result the growth stops. So that is known as contact inhibition. In the stationary phase the growth stops. One reason is this contact inhibition. Got it? Understood? Is it audible or not? Can
Yes,
you
ma'am.
understand?
Yes, ma'am.
So
Yes,
because
madam.
contact inhibition, it prevents the proliferation, it gives a negative signal, okay, inhibitory signal for the growth of the cells. Okay, as a result the proliferation stops. So that is contact inhibition is one reason why the cells doesn't multiply in this stationary phase. Okay, not only that, this stationary phase is used by many scientists to study what is the maximum number of cells that can be grown in the given media. What is the maximum number of cells that can be grown in the given media? To understand that also, scientists, they study this stationary phase. Okay, so the cell growth is limited here. What are different conditions that limit the growth of the cells in the stationary phase? Okay, why growth is stopping? Why the cells are not multiplying here? The factors are nutrient depletion. In the log phase, the cells are multiplying. There are many cells. All the cells, there is a competition and they are taking nutrients and from the media, and in the stationary phase, all the nutrients were exhausted. Nutrient depletion. As a result, that limit the growth of the cells. Okay. Another reason is limited growth space. As I told you, when cells are proliferating, the space in the flask is very limited. There is no space for the new cells which are being formed after division. Then automatically that growth stops. Okay. Not only that, several metabolic products, byproducts, are released into the media and they may be toxic to the cells. That is another reason. Accumulation of metabolic byproducts. So these three are some limitations which inhibit the growth of the cells in the stationary phase. हम बोलते हैं stationary phase में growth नहीं हो रहा है, cells are not proliferating. What are the reasons? क्या क्या reasons है? Because nutrition depletion, the space is limited, right? And accumulation of toxins in the media or metabolic byproducts in the media. So all these are factors that prevent the proliferation of the cells and the cell growth is limited, okay, in the stationary phase. At the end of stationary phase, the cells are in live condition, the cells are active, but they are not proliferating. And you can see some metabolic byproducts are also released in the media. Okay, so after this stationary phase, suppose the cells are not attended, okay, nutrients का depletion हो रहा है, at the same time growth space, you have to split the cells. If we are not attending the cells, if the cells are left unattended, then what happens? Decline phase starts. Means the cells will start dying. Okay. In the stationary phase, it is very important. The cells are active, they are in live condition, but there are some metabolic byproducts are released in the media. Okay. So after stationary phase, as I told you, if the cells are unattended, the next phase starts, that is decline phase, where the cells will start dying. Okay, so that phase is known as decline phase. So there are two different death mechanisms by which the cells die. Okay, one is by necrosis, another is by apoptosis. Okay, so there are by two different mechanisms the cells they die in the decline phase. First one is necrosis. What is this necrosis? It is a passive process. Cells, when they are subjected to the stress conditions, what are the stress conditions? There are no proper nutrients, there is no space for the multiplication, the media, it became toxic and it is filled with several metabolic byproducts. All these conditions are giving stress to the cells. Okay, so it is a passive process, it doesn't require any energy. It normally occurs whenever cells are under stress conditions. Okay, so when cells are under stress conditions, this necrosis process occur normally. It is a passive process. So during this necrosis, what happens? The plasma membrane is broken down. Okay, as a result, what happened? The fluids are entering into the cell. Cell swelling takes place, and finally it leads to the bursting of the cells, right? Cell get ruptured and cells die. You can call this necrosis as extended stationary phase. When cells are left unattended at the end of the stationary phase, the decline phase starts and cells will start dying. The dying of the cells is by two different mechanisms. One is necrosis, which is a passive process, and generally it occurs in the cells which are under stress. Okay, because of the stress conditions, necrosis process starts automatically inside the cell. As a result, what happens? It breaks down the plasma membrane. Cell rupture takes place. Bursting of the cells occurs and cells die. Another process is apoptosis. What is this apoptosis? It is called programmed cell death. It is a suicide mechanism of the cell. Generally, it takes place in vivo conditions, normal physiological conditions, normal aging process में भी cells का death होता है, right? So it takes place in the in vivo conditions, normal physiological conditions में अगर cell का death हो रहा है, that is apoptosis. Stress condition में cells का death हो रहा है, then it is necrosis. Okay, so apoptosis is a suicide mechanism, okay, which happens in the cell cultures. Okay, in vivo conditions, under normal physiological conditions also, you can see this apoptosis. Okay, so sometimes this apoptosis may lead to transformation of the cells. What is transformation? We discussed in the previous classes. Can anyone tell me what is transformation? Anyone? What is transformation? I need some answers. Can anyone tell me what is transformation? What happens in transformation? Yes? Can anyone give answer? No? You forgot everything in this holidays. So what is transformation? Some genetic changes occur which may result in the phenotypic changes also. Genetic changes when they occur inside the cell, right? Genetic information, that is DNA, RNA. When there are any mutations that leads to a transformation, right? Genotype change होगा, phenotype में भी changes आएगा. Then you call that cell as a transformed cell, right? So apoptosis is a genetically programmed pattern of cell events, okay, which may lead to transformation. Okay, what are endonucleases? Can anyone tell me? Endonucleases. Not only
Ma'am,
that, during, yeah,
they
yes.
breaks the DNA from inside.
Yes, very good. So endogenous endonucleases, these are some enzymes which are activated during the process of apoptosis and they cleave the DNA into fragments. So when DNA is ruptured or when the integrity of DNA is lost, the cells die, right? So this apoptosis, this endogenous endonucleases are the enzymes which are activated during the process of apoptosis and they cleave the DNA into fragments.
Ma'am, I had a question. Ma'am, what
Yeah.
is in vivo under normal physiology condition? I didn't
In
got that
vivo
right.
means inside. Suppose you are taking a tissue from any animal. Okay. So within the body conditions, normal aging process में क्या है? Cells will die within the body also. In vivo means inside, within. Okay. In vitro means in the laboratory conditions.
Okay,
Within
ma'am,
the
thank
body,
you.
inside normal physiological conditions, inside the body or inside the living system. Okay.
Ma'am, then necrosis is due to in vitro conditions.
When the cells are under stress, yes. Yeah.
Okay, ma'am.
When the cells are under stress. Okay. So genetically programmed pattern of cellular events, okay, they lead to the transformation of the cells. So even the endonucleases are activated and they cleave the DNA into fragments. Okay. So normally when you are growing cells, you can separate the DNA and you want to know how the cells die. For that, cytotoxicity studies. Suppose I did one work that is using silver nanoparticles. Okay, I want to check the cytotoxic potential of the silver nanoparticles. These silver nanoparticles were synthesized in my lab. Okay, I want to check how the silver nanoparticles are cytotoxic to the cancer cell lines. Okay, so I took one cancer cell line and I seeded in the cells and the cells are growing. Okay, I added the silver nanoparticles and they are cytotoxic to the cell. As a result, some cells they died. Okay, so I want to check whether the cells are dying during the necrosis or due to the silver nanoparticles. I have to rule it out, right? So for that, what I will do, I will separate the DNA of the cells and I will run agarose gel electrophoresis. When I run the DNA, which is separated in agarose gel electrophoresis, I can see the DNA is in the form of fragments. I can see in the form of a ladder. Okay, so that confirms that it is due to the apoptosis. Okay, it is not due to the necrosis. So apoptosis, to rule out whether the cells are dying with necrosis or apoptosis, the only test we can do is to check whether the DNA is intact or it is fragmented. Okay, the fragmented DNA will give you a ladder-like appearance on the gel. When you run the sample in the agarose gel electrophoresis, you can see the fragmented DNA in the form of ladder. Okay, so this is decline phase. Not only that, during apoptosis, the cells will shrink, the microvilli will disappear, the nucleus will condense and it gets fragmented, right? So cells themselves will fragmented with a cellular content inside. Okay, so it is a hallmark of apoptosis. DNA ladder, you can tell it is a hallmark of apoptosis. So fragmentation of the chromosomal DNA into small nucleosomes, okay, of 180 base pairs. If the DNA is fragmented, you can tell that is due to the apoptosis. Okay, so that is the decline phase. The cell death will happen in two different mechanisms. One is necrosis, another one is apoptosis. During apoptosis, what happens? The cells will shrink, microvilli will disappear, the nucleus gets condensed, and it is also fragmented by endogenous nucleases, right? And the cells, they themselves fragmented with cellular content inside. Okay, and fragmented chromosomal DNA you can see. Next is cryopreservation. So these are different phases. Okay, so different phases of the growth. When you are growing cells in the animal cells in culture, you can see all these phases: lag phase, log phase, stationary phase, decline phase. Okay, so let us check the time. What is the time? Sanchari, can I continue? Hello? Hello?
Yes, ma'am.
Hello, Sanchari,
Yes,
can
ma'am.
I
Yeah.
continue?
Yes, ma'am. You are having 4 minutes more. Yeah, ma'am.
Okay.
Yeah.
So next is cryopreservation of the cells. Okay. So after growing cells, when your cells are growing well in the log phase, you want to keep them, store them for the further usage. Okay. So you have to store them. You have to keep stock of your cells. Suppose you are working for 1 month and you are going on leave, then what you will do? You have to store the cells while the cells are multiplying rapidly. During that time, you will store them and keep in the and keep a stock, so that after, when you want to grow the cells again, you can separate them. You can revive and use them easily. Otherwise, the process starts from the isolation of cells, right? Which is very tedious. So once the cells are isolated and you are growing them successfully in the laboratory, you can store them. You have to cryopreserve some liquids of the cell culture. Okay. So for storing, for stock purpose, you will do cryopreservation. So what is this cryopreservation? It is a process where you are preserving the cells in the frozen state. Cryos means frost. Okay, you are storing the cells at very low temperature. Okay, in deep freezers, that is minus 80 degrees centigrade, where we are storing the cells. So in this low temperatures, what happen? All the physical as well as biochemical reactions which are taking place, cellular activities which are taking place in the cell are halted. They are stopped during this cryopreservation process. Okay. So very low temperatures. We use this cryopreservation technique to store the cells, not only cells, even tissues, organs. Okay. What is the temperature of liquid nitrogen? Can anyone tell me? What is the temperature of liquid nitrogen? It is
minus
minus
196
196 degrees centigrade. Okay. We are using this liquid nitrogen for storing the cells. Okay. So cryopreservation means storage of cells, tissues or organs at a very, very low temperatures using liquid nitrogen. Okay. At such low temperatures, what happens? All the physical and biochemical reactions, they will stop. They get halted. Okay. Long-term storage ke liye we can store in this method. But you can ask me a question. When we are freezing cells, okay, when we are freezing cells, so when you freeze, what happens? The ice crystals which are formed may damage the internal structures of the cell. Even the cells are being damaged, right? We want to protect the cells from this during cryopreservation process. For that, we will add some cryoprotectants. Okay. So DMSO is one cryoprotectant, that is dimethyl. What is DMSO? Can anyone tell me? What is DMSO? It is a universal solvent. Anyone? Can they give answer? What is DMSO? Can you see the structure here? DMSO. It consists of two methyl groups which are linked to the sulfur, and sulfur is bonded with the oxygen. So DMSO full form kya hai? Dimethyl sulfoxide. So it is added as a cryoprotectant during cryopreservation of the cells. Okay. So this cryoprotectant will protect the cell during cryopreservation process. So in the next class, we will study what is this cryopreservation, what is the actual mechanism that is happening with the cryoprotectant. Okay, what are different cryoprotectants? What is the process of freezing? We will study, and we will also study about the significance of cryopreservation. Okay, so in the next class, we will study in detail about the cryopreservation.
Ma'am, can
Okay.
you send the PDF in classroom?
Yeah, sure. I will share the notes, PPT, as well as the book which I referred. I will share even that book also. Okay.
Thank you, ma'am.
Okay. So that's the end of today's class. Thank you for listening to me patiently. If you have any doubts, you can ask me. Any doubts? Okay.
No, ma'am.
Okay. Then thank you. Thank you for listening to me patiently. Bye.